Okay, so when we were putting this together, we were going to do another exercise in Galaxy, and unfortunately we don't have time for that, but we decided to just run quickly through the slides for that anyway, because it talks about how we process the data for whole genome DNA sequencing and variant calling, and it helps to understand what's happening when you run the searches. So we're going to run through this quickly. I also want to just highlight here that although we're not going to use it today, in Galaxy we do have configured workflows for variant calling. So just like the ones used for RNA sequencing yesterday, we've got some for variant calling as well. So if you're interested in that, then Omar or Mark or myself would be good people to just show you how where to find those workflows and how to set those up. Okay. And now my mouse isn't advancing things. There we go. Okay, so let's start by thinking about why we want to do whole genome sequencing. So one thing you might want to do is create a reference. If you're studying something that hasn't been studied before, or if the reference you have isn't very good, you might want to create a new one. So that's one reason to do whole genome sequencing. But often the questions are a bit more specific than that. So comparative studies, why is one organism free living whereas a closely related organism is a parasite? Why is this strain virulent and that strain less virulent? Or what happens when you put a species under drug pressure or metabolic pressure? So if, for example, if you have a strain that is resistant to a drug, how and where is it different from a strain that is sensitive to the same drug? And it's also, there are obviously whole fields of study that examine how species diversify and how species are related. And SNP calling is a very important part of those fields of study as well. So when you get whole genome sequencing, there are really two routes you can take with it. So the first of those is to do de novo assembly. So I'm just going to touch on this very briefly. So it's just this one slide because we're not really going to cover it, but it seems to complete this lecture. So all sequencing technologies fragment DNA to some extent. And the idea of de novo assembly is to reconstruct the genome from the fragments, a bit like doing a jigsaw puzzle. And that's the process that you go through to create a new reference genome. So if you're doing this, it's easy to do it with a sequencing technology that generates longer reads, so something like PacBio or Nanopore. And if you think about that conceptually, it's the difference between if you imagine the same jigsaw puzzle, but one's been made for a child and has 6 really big pieces, and the other one's been made with 2,000 very small pieces. It's obviously going to be easier to do the one with 6 big pieces. And using longer reads is basically the same idea. You have bigger pieces and it's easier to figure out how they fit together. So the applications for de novo assembly are often to generate a new reference genome, but it can also be used to assess regions that vary very highly between organisms, like surface antigens or immunoglobulins, because those are so variable that often mapping reads to those regions is difficult. So sometimes doing a de novo assembly of just those regions can be helpful for you to figure out what's going on there. But in this, we're really talking about not so much about creating references, but about these other types of studies that require data about sequence variation. So when we're talking about sequence variation, what we're doing is we're aligning our reads to a reference genome, and we're looking at where the sequence of our aligned reads is different from the genome we're aligning it to. And if you align different samples to a reference genome, you can then ask, okay, so where is this sample the same as the reference genome and this sample different from the reference genome? And that's how you can tell the difference between your samples. So it's important to use a sequencing technology with a relatively low error rate and where the error profile is quite well understood. and that helps you to figure out what's a real mutation in your sample and what's a sequencing error. And for this reason, it's very common to use Illumina for this. So Illumina has a lower error rate than either PacBio or Nanopore, and the error rate and the error profile in Illumina is very well understood. And so we're going to be, well, all the data that we have currently in our databases using looking at SNPs is based on Illumina. So the pipeline, again, like yesterday, I've laid it out on the left. And hopefully this part of it looks very familiar to you, because this is basically exactly the same as what we talked about when we did RNA sequencing yesterday. And so I'm going to whiz through the next few slides because they're actually exactly the same as the ones that were in the RNA-seq talk the other day. I've left them in this slide deck so that if you're coming back to this and you're interested in variant calling, you don't have to go and find the other slides as well. But I'm just going to skip through them very quickly so that we can get on to actually doing some exercises here. So quality control, exactly the same as before using FastQC. Trimming off poor quality 3' reads, exactly the same as before. Again, we can discard adapters at the 3' end, exactly the same as before, and aligning reads to the reference genome. There's one slight difference here, and that is the fact that when we're aligning reads that originate from genomic DNA to a genomic reference genome, we don't have to worry about splicing anymore, because we're not sequencing something where introns will have been spliced out. We should be sequencing something that is just linear. And so we don't need to use these special alignment tools designed for RNA sequencing that understand about splicing. The two most common tools that you'll probably see for aligning whole genome sequencing are BWA and VEuPathDB. Can we mute, please?
Let me see if I can mute them.
Yeah. Thank you. Okay. So once you've aligned your reads to a reference genome, you can just load your alignment into a genome browser and you can start to see variants doing this. So this is an alignment file that has been loaded into JBrowse. So this is a gene at the top, this blue bar, and each of the gray bars is an individual sequencing read aligned to the reference genome. And where you see a colored block, that is a position in a read where the base in the read is not the same as the reference genome, and they're color coded for different bases. And you can see a couple of things here. So over on the left, you can see several blocks which occur in only one read, and they're just kind of randomly scattered around. And those are most probably sequencing errors. Over here, you can see two very obvious examples of SNPs. And these would probably most likely be homozygous SNPs. And you can see that it's very clear here that every read that maps in this position has the same base change at the same location relative to the reference genome. And so we can infer from that that in our sample, this base in the reference genome has changed. And then the last thing to point out here is I've actually got two samples loaded here. So there's one at the top and one at the bottom. And you can see that these two obvious SNPs are occurring in the top sample and not the bottom sample. And so we can use that information to tell us that there's something different about this top sample compared to the bottom sample. But just like yesterday, it's all very well being able to look at these in the genome browser, but how do we find them globally and how do we assess them? So the idea of how a snip caller works, so snip callers are the tools that we use to find these, and they're basically using exactly the same idea as what we just looked at. So I've put a schematic up here, which is basically showing the same thing. So at the top, we have a reference genome, which is this black bar, and the blue bars are individual reads aligned to the reference genome, and the red dots are positions where a SNP is, where a base in the read is not the same as the reference. And there's basically sort of 4 cases happening here. So case E is really easy. all the bases in all the reads here are the same as the reference genome. So that's fairly easy to interpret. There's no SNP there. Case A is the opposite, and it's basically the same as we were just looking at in JBrowse, where all or nearly all of the reads at this position have a mutation in relative to the reference genome, and it's the same mutation in all of the bases, in all of the reads, sorry. And that we would probably interpret as a homozygous SNP. In cases B and C, we've got something going on where approximately half of the reads that map to this location have a change in them relative to the reference. And in this case, this is probably a heterozygous SNP on a diploid genome. So in that case, what's happening is you've got two copies of the chromosome, and you've got a mutation on one of those copies, but not the other copy. And so you're sampling equally from the two copies when you do your library prep. And so half of the reads will have that mutation from one copy, and the other half of the reads will come from the other copy of the genome without the mutation. And so you see this, we call it an allele frequency, where roughly 50% of the reads are differing from the reference. And then in the last case, we saw some of these in the JBrowse as well. We have just one base that's different from the reference. And that is most probably a sequencing error. It could also be a case that you have one individual in a population that has a rare mutation. But most likely in this case, it's probably a sequencing error. So some things to think about here. First of all, these ratios assume that this sample is either a culture, a clonal culture, or a sample from an individual. So you're assuming that everything in the sample is the same. If you're sampling a population, then these ratios don't work out, because obviously you don't know how many individuals in the population are likely to have a particular variant. The other thing is about this case D, where we have just one SNP. If you have a population and you're looking for rare variants, something just to be a little bit aware of is that Illumina has an accuracy of 90%. And so if your variant is rarer than that, it's very difficult to tell the difference between a real variant and a sequencing error. And at that point, you're really into, if you really think that this might be interesting, you're really into taking that away and validating it somehow. Because you get to the point where you just can't tell the difference between real mutations and errors. So some automated tools to call SNPs here. BCF tools is a really good, quite basic SNP caller. GATK and Freebase are also very good. They're slightly more sophisticated, and these use Bayesian modeling to take into account more information about things like the mapping quality and the location of the SNPs in the reads and what other features are around the SNPs in order to improve the probabilities of the SNP calling. But any of these three SNP callers are very good to use. So as well as looking at SNPs, the other thing you can get from whole genome sequencing is information about copy number variation. We can get that by exploring the coverage. So when you do whole genome sequencing, the expectation is that the coverage should be even across the genome. And that's because this isn't RNA sequencing. You should have, if you have a copy, if you have a single copy gene and you have two copies of the chromosome, you should have exactly the same amount of input material as you would do for another single copy of gene on two copies of the chromosome. So if you're sampling evenly, the coverage should be even. In reality, it's not quite that simple. So we see some local variation associated with GC content. And that happens because most library preps include a PCR or an amplification step, and GC-rich regions tend to amplify better than AT-rich regions. And we also see some variation associated with repetitive or very variable regions in the genome. And that can happen because it's difficult to align reads back to those regions in the genome. So you quite often see that the coverage in those regions is either very noisy or it just drops. And we'll have a look at some examples of these in a minute. But changes in coverage can also tell us about copy number variations. And the way this works is if you imagine that this is a sample, this is the reference, this is the isolate you've sampled at the bottom, and a block of the genome has been duplicated. And because this is whole genome sequencing, we're sampling evenly all the way along this. So this is what our input looks like. But this is a duplication relative to the reference genome. So the reference genome only has one copy of this region. So the reference genome looks like this. So just one copy of this red region. And what happens here is that reads that map outside that region are fine. They can map just to where they would normally. But the green reads that map to the first copy of the duplicate in your sample, that originate from the first copy of the duplicate in your sample, and the pink reads that originate from the second copy of the duplicate in your sample, will all map back to the single copy of that region in the reference genome. And so what you see is that the coverage is fairly even, and then it goes up, and actually you would expect it to more or less double here for a duplicate, and then it drops down again. And again, we'll look at a real example of this on another slide in a second. So this is Plasmodium falciparum, chromosome 1. And what we're looking at here, this is from a desktop genome viewer called Artemis. But what we're looking at here is 4 different samples in 4 different colors. And we're looking at the coverage of reads aligning to the genome. So some things to highlight here. This big block in the middle is what I was saying that we should expect, that the coverage should be more or less even. You can also see what I was talking about here. You see how it is broadly even, but on a small scale, it goes up and down a lot. And that's very likely related to the GC content of the genomes. On the left-hand end of the chromosome, we have, first of all, a very repetitive region, followed by the region that encodes a lot of riffins. And so you can see here that at this end, the coverage is quite noisy in the repetitive regions. It goes up and down a lot. And then in the variable region, it just drops. And that's because we're having difficulty mapping reads back to this region because the genes change a lot in response to immune pressure. But over here, what you can see is that in 2 of the 4 samples, there is a copy number variation. And you can see that it's actually a slightly different copy number in variation in the 2 samples. So the red one is slightly to the left of the blue one, but that they're overlapping. And some features of this that tells me that this is a real copy number variation compared to noise like this is, first of all, these very sharp edges to the coverage increase. And secondly, that although you can't see the scale on here, the magnitude of this is a direct multiple of the background. So in this case, this is actually a triplicated region. And I know that because the mean up here is 3 times the mean down here. So we can use these to find copy number variations in genomes. So the last thing quickly to talk about with SNPs is that we also need to do something called annotating them. So what I talked about before was finding SNPs in a genome and how you'd go about doing that. But the pipeline I talked about so far only gives you information about where the SNPs are. And so often the more interesting information is what are those SNPs actually doing? So are they in a coding region or not? If they're in a coding region, are they synonymous or are they non-synonymous? So does the protein change? If the protein is changing, is it just one amino acid that's changing, or have we introduced a stop codon and truncated it entirely? If we've changed amino acid, is it something relatively conservative, like a leucine to an isoleucine, or have we done something like changed a glycine to a phenylalanine, which would have a much bigger effect? So we also do something called variant annotation, and there are several different tools that can do this as well. These are some that might be of interest. So the variant effect predictor is a really good tool run by Ensemble. We have SNPF available in Galaxy, and Anavar is another one that's good to look at. So just lastly, as I said at the beginning, although we're not going to get a chance to look at it in this workshop, we do have a Galaxy workflow that's pre-configured for you to use. And just like yesterday, this is the layout of our workflow, and you can see how that lines up with the pipeline that I've just talked through here. So if anyone wants to know more about that workflow, then please grab one of us in the next session. So that's all I wanted to talk about. And then I think Omar was just going to run through some of the searches. I'll just stop sharing a second, but there was, I'm trying to stop sharing. There we go. There's a quick question in the chat, which was, how does heterozygosity and homozygosity affect the CNV analysis? And I think my answer there is that it depends on what you're trying to do. So it doesn't matter if your genome is homozygous or heterozygous. You still see a coverage increase where you have a copy number increase, and you'd still expect that to be a multiplier of the background. Where it differs is when you're trying to predict how many copies of a gene or of a section of the chromosome there actually are, because obviously your background is different. So in a homozygous genome, your background would represent a single copy. Sorry, in a haploid genome. And in a diploid genome, your background is likely to represent 2 copies. So when you're looking at the difference in magnitude, you have to remember what the background actually is as well. Does that make sense?
Great, thanks, Catherine. And obviously, as usual, the lecture is being recorded and will be made available probably by tonight or tomorrow sometime, and you'll be able to review it as soon as possible. So what I'm going to do quickly is just show you some of the parameters that are in our SNP and variant CNV searches, and those are basically going to be the ones that your searches are, or this next exercise is based on. So I'm looking at PlasmoDB, but obviously this can be on any of the sites where we have this kind of data. Obviously, we don't have it for all of our organisms. And again, as usual, if you're looking on GRDDB and you're like, oh wow, I know there's all these isolates that have been sequenced and they're sitting in GenBank. Why don't they have them? Just let us know, and we'll go ahead and load them. So you'll notice here in the searches on the left, most of the searches that we've been doing so far have been gene focused. And so if you open this up, we have gene focused searches. And in here, in your gene focused searches, we have genetic variation searches. So exactly what Catherine was saying, that when you're looking at SNPs, you may be interested in identifying SNPs in general in a genome that distinguish a population of Plasmodium isolates from East Africa and those in West Africa, for example. And you're just looking for polymorphisms because maybe you want to design a PCR assay that will distinguish isolates from those 2 regions. In that case, you may not care that those are within a gene or not. It doesn't really matter, because all you're looking for is distinguishing mutations or polymorphisms. However, in many cases, you're really curious about, well, I want to know if my mutation or my polymorphism is within a gene, and furthermore, I want to know what is the effect of that mutation. And we do this in our databases, and we allow you to do that, and I'll demonstrate in a second. So, but in that case, you would be running searches on genes, so looking for polymorphisms within genes. However, as I mentioned before, you could also just run SNP searches and distinguish between SNPs between 2 different groups of isolates or within a group of isolates. And I will go there maybe first and then switch to the gene searches. So let me pick the difference between 2 isolates. And you notice here at the top, you have set A isolates. And then as I scroll down, you have set B isolates, right? So you have to decide, you're again the one who's asking the question. So you have to decide which isolates you want to compare. And we load whatever metadata or information about the isolates we have, we load that in these categories here. So for example, you can select by country. I can say, well, I want all the isolates from Brazil. And in my set B, I want to compare them to the isolates from, I don't know, we can pick Kenya, for example, or actually the bad example, there's only one there. Let's pick Mali, they're 23, for example. And so that would be my comparison, my samples I want to compare to each other. But for each of them, you have to define some characteristics, right? And so for set A, you can say, well, I want to define the frequency. I want to define the major allele frequency or minor allele frequency, depending how you look at this, and also the percent isolates with a base call, okay? The frequency is, Catherine was talking about that before, and this helps you define whether a polymorphism is haploid, diploid, or homozygous, heterozygous, or more, right? And so in this case, you would be, when it's set to 80%, you're saying, well, I want to return any, for each organism I've selected, I want to return any SNP that has a read frequency of greater than 80%. Okay, so that means that in a perfect world, obviously, if something is homozygous, then 100% of the reads, the frequency of 100% would match that particular allele, right? And so we set it to 80 and above because it's never perfect. So we set it to 80 and above. But as you can see here, you can set, between 20 and 80%. So you can look for a variety of types of allele frequencies in your search. So if your organism is a haploid organism, so in most cases you will keep this at the frequency of 80% or higher. But it's possible that even in a haploid organism, you may be looking for duplicated genes and looking for alleles that are higher than a homozygous situation. The allele frequency, again, just as a reminder that any of the parameters we have on our sites have a little question mark next to them. And if you mouse over that question mark, you get a lot more information. And also for many of our searches, if you scroll all the way to the bottom, we have even more information. So just right now, what I just told you about the read frequency, I probably should have showed this when I was telling you about this, but you can see here and see the example. So this would be 100% read frequency would match this. Or in this case, this would be a 40%, I believe, read frequency and so forth. Okay, and so you can read this. There's a lot of detail here and information. With the minor or major allele, basically, if you have 10 alleles, right, and 6 of them are of a particular allele and 4 are another allele, like 6 of them are an A and 4 are a G, right, then your major allele is the A and the minor allele is the G. And this helps you, again, define, potentially, this allows you to search for alleles that are rare. So you may want to look in a population for something that has a minor allele frequency, and that may define maybe, in an ideal world, if you have enough sampling, that may help you define emergence of new drug resistance or other traits that you may be interested in. The percent isolates with a base call. So this is a metric that allows you to increase the stringency of your search. And so if you look all the way in the bottom here, we have right here. So for example, at this position here, there is no sequence for many of the reads. Only 4 of the reads had sequence, the top 2 and the bottom 2. But all of these, there was no sequence there. The sequencer could not call the read, the nucleotide at this position, or the reads terminated here, and this is just a gap in the sequencing. But the bottom line is that this is below the threshold which we use, which our programs use, which is less than 5 reads, that below which we do not call a SNP in that position. We basically say that we can't be confident about calling anything here, so we're not going to tell you, we're not going to call a SNP here. And so this percent isolates with a SNP call at any position allows you to increase this your stringency. So if I had 10 isolates selected up here, and I'm saying that I want at least 20% of them, more than, or 20% or more, to have a base call at a position, then I'm saying that at least 2 of them should have a base call before I return that position in my results. I could increase this and say, well, I'm going to make it 100, in which case you would be looking for SNPs that have sequence or alleles, locations that have sequence from every single read every single sample that has been sequenced in the experiments that we have in the database. Okay, so I'm going to switch to back and look at the SNPs for the gene. So I'm looking at the gene searches, and I'm going to look at the SNP characteristics. And you will notice, again, same thing, you can select the samples. And in this case, the question you're asking is, I want to find genes that contain SNPs of the following characteristics, right? So I'm going to find genes that have SNPs that come from isolates that are, you could select all of them that are from any sequenced strain in the database. And now you'll notice here that you're defining similar characteristics as before. So I won't go over read frequency again or minor or major allele. I usually leave the minor allele at 0 for most of my searches. Basically, I don't care. And also the percent isolate with base calls, that one will help you increase the stringency. But now here, you can actually define the type of SNP that you want to return, or the type of the genes that contain a particular type of SNP. So in this case, I can, for example, say, I want to find all genes that have non-synonymous SNPs. And you can even define the number of the non-synonymous SNPs that you would like those genes to contain. And for example, I can change this to 10, right? And so in this case, any gene that has at least 10 non-synonymous SNPs in the isolates that I selected will be returned in my searches. And you'll notice here, there are many other parameters, that you can play around with, because as you can imagine, a really big gene may have lots of SNPs, but and a really small gene may appear to have a small number of SNPs, but ratio-wise, based on SNPs per gene length, they may actually have the same frequency or percentage of SNPs across the gene, right? And so this may be an important metric or an interesting metric that you want to play around with. And so there are many other parameters here. I won't go through all of them. Hopefully they're self-explanatory, but there's obviously a lot more information here that'll help you define things. Okay, going back to the homepage, just to point out that we do get most of our SNPs come from whole sequencing projects, but we do have SNPs from other types of assays as well. For example, we have SNPs that come from a chip. This is just like a microarray, basically, where you're defining the difference. The sensitivity is basically based on the polymorphism, basically, and you can tell essentially in a microarray environment, looking at changes in intensity as to whether there is a SNP at that position. And the array is obviously plated with known, a large, not necessarily a large number, but a number of SNPs that are known in a particular region, and then that allows you to sort of distinguish the types of SNPs from each other. But the search runs pretty much in the same way, in the same manner. You can select the characteristics of the SNPs. And then you can also ask these copy number variation questions. And so we have two types of searches. One of them is it's called just the copy number, and then one of them is called a copy number comparison. So let's start with the first one. So the copy number variation. And again, you can select any of the resequenced isolates that we have. And then you have really two parameters that you can select. And again, mouse over these and read these carefully. There's a lot of information here, but you can select basically, you want to return the gene whose median copy number across the selected isolates meets the criteria, right? Or you can basically make it by selected isolates, by one of the selected isolates, right? So you're saying, well, I don't care, as long as one of the isolates I select has a CNV at that position. The other metric that you can choose, it's the copy number metric, and you can decide as to whether you want to find these based on haploid number or based on gene dose, right? So you can change this from haploid number to gene dose. And again, described here, but you can see here that the haploid number is really the number of copies within a single chromosome, and the gene dose reflects the total number of copies. So it takes into account ploidy of the chromosomes and so forth. And there's a really nice example. Basically, if you have a single copy gene on a diploid chromosome, it has a haploid number of 1 and a gene dose of 2. Okay. And then this is the operator. You're basically saying, well, I want the haploid number to be greater or equal to 2, or you can change it to any number that you wish, obviously. Okay. So The second type of copy number variation search is a comparison search, and this allows you to basically compare with the reference, basically. And so so again, you can select the median or the selected isolates. Okay, so when at least one of the strains has the copy number variation. But then here, the comparison that you're asking is compared to the reference, right? So you want it to be either greater than, equal to, or less than the reference genome, right? Okay, so it's already after 11, so I think we probably should move on to the exercises so you can actually play around with them. Let me see if there's a question. And Catherine, was there anything else you wanted to add that I potentially missed?
Sorry, you just caught me with a mouthful. No,
Not
I
a
think
problem.
I don't think I've got anything particular to add.
Okay, and I see a chat in the window that there is, in the next question, there were some incorrect answers in the exercise. So some of the screenshots are wrong, but Suzanne has applied or put up the correct values in the Slack channel for session 12. So go ahead and check that.
Those are the values I got today. So.
Okay. All right. Cool. So with that, I think we should go ahead and break up into our breakout rooms. Rachel, you can go ahead and open them.